Okay, this video is on a phenomenon called epistasis. Again, what can I say? It's not on your syllabus. Again, it has come up. So, what is epistasis? So, epistasis is where one gene influences another. So it influences how that gene is expressed. It's quite a sort of, it's a lovely thing to ask about, I think, as an examiner, because you're sort of bringing in dihybrid crosses because you've got two genes. They're influencing the same thing. So I think a couple of years ago there's a question, I'm sure it's on Moodle, about Labrador coat colour, and there were black Labradors and yellow Labradors and chocolate Labradors, and it depended on what allele they got for coat colour and what allele they got for skin colour. So the sort of mixture of the genes was influencing. It's also quite a good way that they can ask about sort of little enzyme pathways, and again in a longer question than this one, they could bring in a bit of synoptic perhaps about transcription and translation or enzyme function. There's another great question somewhere on Himalayan rabbits, and Siamese cats are the same, where the high temperature sort of prevents body temperature prevents the allele being, the enzyme working, and so they get white fur where their body's nice and warm, and at the cool bits at the ends of their noses and the tips of their tails, think about Siamese cat, their paws, then the enzyme that creates the black fur colour does work. And there is definitely a super duper question where they show this by shaving a rabbit, which always makes me laugh. Anyway, there we go. I thought that even may be on YouTube somewhere. I think that might be one of the first things I did. So we're going to sort of look at this as an illustration of this sort of one gene, one enzyme thing going on, with a little pathway and with an epistatic. You can see we've got a little space here to do a cross at the end. So, just reading through the question then. The inheritance of colour in sweet peas is controlled by two pairs of alleles. So this is a dihybrid cross. We've got two pairs of alleles. We've got big A and little a, and big B and little b at two different gene loci. So you can assume that they're on different chromosomes. The diagram shows how the dominant alleles determine flower colour by controlling the synthesis of so these are the dominant ones, controlling the synthesis of a purple pigment. So allele capital A makes enzyme A. So in between there, of course, you're going to have transcription, translation, modification of proteins. And it changes a white precursor substance into an intermediate substance, also white. And then we've got a second locus with allele B on it, making enzyme B, transcription, translation, all the rest. And that converts the intermediate substance into a purple pigment. So, thinking about enzymes, this is acting on this. This is the substrate, and that's the product. And for enzyme 2, this will be the substrate, and the product is this purple colour. So to get a purple colour we need at least one allele A and at least one allele big B. So what does it ask us? Plants with the genotypes little a, big B, little a, big B, little b, little a, little b, little b. So the thing in common that they've got is the little a. All have white flowers. Using information in the diagram again to explain why plants homozygous for this mutant allele A have white flowers. So what can we tell from the diagram? Well, we know that the precursor is white, and if we've got two little a's, we haven't got enzyme A. So that's one of the first things that we can say. So little a plants, peas, sweet peas. I'm just going to call them plants. Cannot produce enzyme A. The precursor, might as well use the words in the question, that's on the diagram, using information from the diagram. The precursor substance cannot be converted into the intermediate substance. So if we haven't got this enzyme here, then that's not going to be made into that. I think we need to talk a bit about this capital B allele here. Even if enzyme B can be made, so if we've got allele big B or big B little b, we can still make that enzyme, tick, we can do that, but we haven't got any substrate to act on. There will be no substrate for it to act on to make purple pigment. So flowers are all white. Excellent. So that's just about the relationship between the enzyme and the allele. Use a genetic diagram to explain the genotypes and expected ratio of phenotypes when plants with the genotypes big A, little b, and little a, big B are crossed. So obviously we're looking at dihybrid now. I've been given the genotypes, that's great, that just means I need to foil this. So, genotypes of gametes, first letter, big A, little b. Outside, big A, little b, got one. Inside, little a, little b. Last, little a, little b, I've got one. Crossed with, I'm going to put my cross in so I don't pick up here. First, little a, big B. Outside, little a, little b. Inside, little a, big B, I've got one. Last, little a, little b, I've got one. Well, not left me a huge amount of room to write my Punnett square. Cheers. Cheers, examiner. Good old AQA. So, writing very small now. Big A, little b. Little a, big B. That's parent 2. So, genotypes of offspring. Big A, big B, little b. Big A, big B, little b. So back to my diagram. So we can make the intermediate, we've got big A, and we can make the purple pigment purple. Little a, big B, little b. That's going to be white. That's one of my genotypes up here, isn't it? Big A, little b, little b. Okay, more complicated. Can make enzyme A, can convert precursor to intermediate, but no little b enzyme to make purple, so that will also be white. Big A, little b, little b, white. And last but not least, little a, little b, and that will also be white. So this is actually now starting to look fairly tricky, isn't it? Because now we've got a 3-to-1 ratio of, but it's white to purple. So our recessive is looking like, you know, it's giving that sort of dominancy kind of thing, isn't it? So I'm still going to write it the right way around though, 3-to-1, but I need to make sure that I put white to purple, because that relates to the 3 and that relates to the 1. It's kind of the backwards way, isn't it, of the normal, what we would expect in a monohybrid cross. So, super duper. And notice that we've got a 3-to-1 ratio because of the influence of one allele on the other, whereas normally we'd be looking at a 1-to-1-to-1 ratio with that kind of cross in a dihybrid situation. But because of one allele is influencing another with regards to colour, we're getting just the 2 phenotypes. So do watch out for those. You follow the steps, you follow the logic, you foil, you do what you're told, you read the information. You're never ever going to make a mistake in genetics. Genetics is such a lovely subject.